Okay, so here we're going to begin our discussion of cellular respiration, and we're going to be looking at aerobic respiration. The beginning of this process, we're going to begin with a molecule of glucose. So we talked about how glucose is the most common substrate for ATP production. Well, I've drawn the glucose molecule for you here, right? It's C6H12O6. That's a molecule of glucose. Now, the reason I have the hydrogen in red is because that's where the energy is stored. The energy associated with this glucose molecule is stored in the electrons contained within the hydrogen. The carbon and the oxygen, we're simply ultimately going to get rid of through this process. Ultimately, the carbon and the oxygen will become carbon dioxide. And what do we do with carbon dioxide? Yeah, that's just a waste product. We're just going to exhale that, right? We're going to get rid of that. So we're going to remove carbon from the glucose and convert it into carbon dioxide. So the only thing that's left then to actually extract energy from is the hydrogen. So we're going to remove hydrogen from glucose. Anytime you remove hydrogen from an organic molecule, that's called an oxidation. So that's why this is called the oxidation of glucose, because we are removing the hydrogen. But here's the deal. This process of aerobic respiration takes an enormous number of reactions. It takes a lot of reactions. It's going to take 4 primary steps, which we're going to be breaking down each one individually. We're going to see glycolysis. We're going to see the oxidation of pyruvate. We're going to see the Krebs cycle, and finally electron transport chain. But here's the deal. As we remove hydrogen from glucose, that's ultimately the energy source we're going to use to produce ATP. However, remember I said earlier, we cannot remove hydrogen from something, so we can't oxidize something without reducing something else, right? We have to transfer this hydrogen to something else in the interim for a short period of time until we can convert that electron energy into ATP. So in respiration, we have 2 hydrogen acceptors. So we have 2 molecules that we call hydrogen acceptors, that when we pull hydrogen off the glucose, we will actually give the hydrogen to one of these 2 hydrogen acceptors. So the 2 hydrogen acceptors that are used in aerobic respiration are NAD and FAD. And NAD and FAD both will accept hydrogen 2 at a time. So they will actually accept 2 hydrogen at a time. When NAD accepts 2 hydrogen, it becomes NADH plus H plus. That's how that reaction will run. And when FAD accepts those 2 hydrogen, it will become FADH2. So these molecules are simply temporarily holding onto those hydrogen atoms until we will ultimately convert the energy associated with those hydrogen into ATP. And that will take place at the electron transport chain, which is the final step in aerobic respiration. So what we've done here with both of these reactions, these both represent reduction reactions, right? Those are both reduction reactions. We have reduced NAD and we have reduced FAD, because anytime something accepts hydrogen, that's a reduction. A reduction is a gain in hydrogen or a gain of electrons. So, all right, so that's the very beginning process in aerobic respiration.